Okay, good morning, good afternoon, whatever applies to you, and welcome to our first webinar 2022. It is my pleasure today to introduce this webinar dedicated to innovative applications that can be easily transferred into our departments. The first speaker is Dr. Jungbluth from New York, and he was not present online to our first broadcasting today because it was in the middle of the night where he's based. Therefore, at the end of his presentation, there is a session, a pre-recorded session with questions and answers that I had prepared myself. You are more than welcome to ask your questions by writing them as soon as they come up to your mind, typing them as messages into the chat space. They will be collected and answered directly at the end of this second presentation, when both our guests will be present for our live questions and answers session. And now it is time for me to stop and let this presentation start.
I've never taught my students anything. I just try to put them in the best conditions to learn. Albert Einstein. Dept is a leading company offering a complete range of innovative instruments, reagents and consumables for anatomic pathology. Dept is based in Italy, near Milan, with a network all over the world. As you know, Dept is approved CE use provider by NSH, National Society for Histotechnology. If you're interested in gaining CE use by NSH, you're welcome. You're invited to follow the instructions under the website www.dept.com/webinars-list. Before starting the webinar, just a few practical information. The webinar is about 30 minutes. Then we are going to have a question and answer session at the very end of the webinar. So you are kindly invited to have your questions at the very end, typing the questions in the chat for the final open discussion.
Good morning, good afternoon, whatever applies to you. My name is Dr. Jungbluth. I'm working at Memorial Sloan-Kettering Cancer Center in New York City, and I'd like to first of all say thank you to Dept and the whole team, especially Dr. Savio, for having me for this presentation. As you can see from the first slide, I'm the director of Immunohistochemistry. I'll deal mostly with immunohistochemical assays and the setup of those assays. We have a very high volume of novel IHC protocols, and at one point the department decided that this needs to be done in a separate lab by a separate person. And by doing so, we came up with an idea which was actually evolved by the work that I was doing together with my senior tech Denise, who's not on this on this presentation, but we try to improve the high volume of throughput that we have in our lab and a convenient and reproducible way to test new reagents. And the idea that we came up with is called carrier-based multi-tissue blocks. We call them CBMTBs. Most likely the term tissue microarray crosses your mind, but this is a different concept really, and it's not micro as you will see. And yes, it's a novel way how to construct multi-tissue blocks. Let me show something about my background and why I was actually asked to run this lab. I'm a pathologist by training, but that was a long time ago. And since the 90s, actually, I was working in the Ludwig Institute for Cancer Research, and that was an institution that primarily dealt with tumor immunology. Now tumor immunology is in everybody's mind, and it's very popular. The PD-L1 treatments are immunologic treatments. If you think cancer vaccines in general are very popular these days. And I worked in an institution where we did exactly that. And as a pathologist, I covered all the pathology that came up in this lab. And one big arm of what we were doing was also therapeutic antibodies, but also diagnostic antibodies. For a lot of cancer antigens, you really have to know where are they present. So by doing tumor immunology, we were sort of also forced to do diagnostic work and checking where certain tumor proteins, antigens are expressed, because you want to know how they're expressed if you design a vaccine against those antigens. And some of the antibodies that were generated in our lab you actually use because they have been commercialized. One of them is, for example, the melanA antibody, the clone A103. There's a tyrosinase antibody, T311, and there's one to a group of special antigens called cancer testis antigen that was also commercialized. There are many more antibodies that were commercialized, but I just want to let you know I have quite some background testing novel antibodies. So the current position in the Department of Pathology here has a high throughput of requests for new IHC protocols. And, you know, we're one of the biggest cancer research institutions and hospitals in the world, and we have a very active research arm. So one of the key questions is, a lot of times we have researchers that know, oh, you know, there is a new antigen that plays a role in liver cancer, and I think you should get a protocol for this because it has some, you know, prognostic impact and so forth. So we're dealing with a lot of requests from protocols that are completely novel. Nobody has ever worked on them. We have an active research group that deals with them. And on top of it, we have our own pathologists. We over 110 pathologists in our department who are very active, follow the literature and say, oh, there's a new diagnostic antigen, so let's test for this. So we have maybe more, that's a not so new slide, 300 IHC protocols in our clinical diagnostic lab. And we are constantly testing new protocols. So we have to comply, of course, with regulations, nothing without regulations. They're called CLIA regulations here. We have to fulfill certain requirements in order to make commercially available research antibody a clinical diagnostic reagent. So there is no question that we need an efficient way to test new antibodies if they're really specific. I hate the term specific, but I will use it here. But when I use it in quotation marks. What is specificity? It's a more confusing term, but for me, an antibody that's specific is an antibody that detects its epitope. Where the epitope is distributed is a different question. So some people confuse this. So anyways, so we need an efficient way to come up and deal with the demand of new IHC protocols. So we do not generate our own antibodies anymore as we used to do in research. We basically do 99% commercially available antibodies. So when I did the original work in developing antibodies, you know, we had a very good idea about the expression of certain antigens in different tissues. And today, even more so than many years ago, it's almost easier and more reliable to find mRNA expression data than IHC data, because IHC data are prone to, you know, the problems of specificity of the antibody. And for most of the antigens and proteins that are distributed in different tissues, you get very good and reliable mRNA data. One, for example, is the Human Protein Atlas. There are other databases, and I have copied them on this slide, and you can see there are two examples. One is melanA, of course, because we worked on this, and the other one is BCMA, an antigen I really didn't know too much about, but it's mainly expressed in a certain type of lymphoid cells. And it's not so important. I just want to point you to the right side of the slides, which show the expression data for both of these antigens, melon A on the very right side and BCMA, it's also called TNF receptor superfamily 17 on the left side. And when you look at this, I hope you can see my cursor. On the left side of those things is actually mRNA data, and to the right side, the Human Protein Atlas usually displays protein data, mostly based on their own polyclonal. So for me, important is the left side, and we see that this is a very peculiar expression pattern. So looking at this, there's not a single tumor tissue in it. These are all normal tissues. I concluded that maybe the best way to have a fresh look at a new antibody is to look at normal tissues. Well, other people knew this before, and one of the ways to test several normal or several tissues, doesn't be normal tissues, can be tumor tissues, are multi-tissue blocks. And the way you see it here is the way many people do it. They punch it out with the circular punches, or called derm punches, and then they place it into one block and section it. And you see you have actually to take quite a big diameter. They're arranged in a kind of fashion that just happens to be convenient when you embed it. The tissue, the number of tissues you can embed is kind of limited. So when you look at the slides, this is how it looks. The right side shows the higher magnification, and as I say, it's basically random how the tissues are embedded. Embedding more than 5 tissues is tricky. The technician has to hold them in place when the paraffin is poured over it, and so forth. So we thought there should be a better way to do it. So the idea comes as following: why don't we use a carrier tissue? So the idea is very simple. You have 2 blocks. This should symbolize the paraffin blocks that you have, and one is a donor block and one is an acceptor block. So you take an acceptor tissue, and I will talk about which tissues are ideal. We usually take spleen. So the acceptor block is usually spleen tissue, can be other tissues as well. And the donor block is whatever tissue you want to insert into the acceptor block. And basically you punch out a small piece of the tissue. Actually, ideally, this is not a good picture. The cylinder should go all the depth of the tissue, the donor block, and then you insert it into the acceptor block. And then you repeat it. You take a different donor tissue and also accept this in a block. And here is an example how we do this. This is a spleen tissue, which we use as the carrier or acceptor tissue. And in this case, we first punch out the desired circular spleen tissue. In this case, it's 12 millimeters in diameter. Put it into a mold and re-embed it. Means we'll heat this again, then pour paraffin over it, let it grow cold, and you see a beautiful new circular acceptor tissue or carrier tissue. This will serve as the carrier for the other tissues that we're going to insert. And now you see on the left side how we proceed. So you take one of those derm punches that you see, this white one, you get them in all different sizes, and punch out, extract a core. So the diameters are much smaller than what you see on the conventional multi-tissue block, but they are much bigger than a tissue microarray, which is usually limited to 1 millimeter or even less. So these are, you will see on the following slide, 2 to 3 to 4 millimeter in size. The next slide shows you a colon tissue embedded in an upright position, of course, and we extract this tissue with the same size of derm punch. And here we took the punch out so you can actually see it's inside. Normally it remains inside. And then we basically fill this punch into this hole that we made before by extracting one core from the from the acceptor block. And then we repeat this over and over. But you see, this doesn't really look very nice. The following slide shows you even more. If I would show you a high magnification, this would look pretty crappy. And if you look from the side, you know, they're all crooked. They're in different levels. So what is important? You have to re-embed the tissue. What means re-embedding? It simply means that you have to heat the paraffin block and let the paraffin melt completely. If you're a technician listening to this, it's very important that you keep the block in an oven for a long time, let's say an hour or two. Also, what we do when we put them in the oven, technically we put a mold, one of those metal molds, on top of this block and then turn it around, so that this upper side is actually on the bottom of the mold. Then we heat it. And once everything is melted and we are sure that the paraffin also inside, all the different punches and so on is melted, we just place it on one of those, you know, embedding stations, pour a little bit more paraffin over it, put a new cassette on top, and then put it in the fridge. We do not put it on the cold plate, and we do not put it in a freezer. Just put it and let it get cold in a fridge, because it's a much more even process than having it on a very cold cooler plate. And what comes out of all of this is what you see on the left side. This is a re-embedded block. This is the same block that you see up here, just everything in one plane and everything one piece. Why do we do this? First of all, this is not easy to cut. But what you will see is because paraffin is a substance that is based on its ability to polymerize. So when it's liquid, it's liquid. When it solidifies, it gives an even consistency across different tissues. So when you use and place this in a microtome and cut sections, this multi-tissue block will cut like one piece. It's not like cutting a TMA when you ever section a TMA. It feels like you're cutting many, many different small tissues. This cuts like one piece. And this is how an H&E looks from such a block. So this is round. The diameter is 12 millimeters. So I alluded to the carrier tissues before. We prefer spleen for technical or several reasons, but we'd also tried lung, kidney, liver. They all work fine. They just have to be very homogeneous. So what test tissues do we put in them? And this is the tissues that we normally take. You may prefer others. You can insert whatever you like. You should also know that we do not strictly distinguish between test and carrier tissues, because the nice thing, and one of the biggest advantages of our multi-blocks, is that the spleen tissue or any other carrier tissue serves as a test tissue itself. Let me demonstrate you how this works. You can also design a shape, whatever you like. You can make them smaller, you can make them bigger. When we do our initial test of antibodies, we do not use 12 millimeter diameter shaped carrier tissues. We use much bigger ones because it's the only tissue that's going to be on the slide when we test an initial antibody. Now, when we look at the IHC, now this is again the multi-tissue block that we did before. And to the right side, you see it shows a diameter of 12 millimeter and 12 millimeter in overall, and you see that the biggest tissues are the skin and the colon. The others have smaller diameters. Now, the advantage of normal tissues is that the expression of any antigen, and that's important for setting up a new antibody, is usually very homogeneous throughout the tissue. In tumors, this is completely different. For example, if you would take a melanoma to test a new melanoma antibody, it could happen that you find that you're using in the first instance the melanoma as a control tissue that's completely negative for melanoma, which happens in 5% of the cases. And the same applies for other tumor antigens in other tumors. But if you test melanoma on a normal skin, whether it's 2 millimeters, 3 millimeters, or 30 centimeters, the number of positive melanocytes is pretty much the same. And you will see normal melanocytes there. And if you see this in this multi-sample positive, you know that the assay worked. And I think what's also evident is the advantage of the carrier tissue here. We have a much smaller diameter than the tissues in the conventional multi-tissue block, which I showed you before. But they are kept in place by the surrounding spleen tissue, which serve as a scaffolding. So the scaffolding allows for a smaller size and diameter of the tissues, because even when you melt the tissue during re-embedding, the colon and the skin is held in place, and they will not somehow tip to the side, and all of a sudden you don't see any epidermis anymore, or colon. So the way you embed it, it's the way you're going to see this here. Now let's look at the IHC. So one good example is if you check vimentin, for example, you see that they're staying all over the place. Of course, because the spleen is primarily a mesenchymal organ, so vimentin will be positive. And you see one of the advantages is that the space between those punches is not lost space like in a conventional block, but it's actually spleen tissue, and the spleen tissue will serve you as a test tissue, just like the other tissues that were inserted. Now, how about the high resolution when you look at this? And this is, again, vimentin, and look at this. On the left side, you see the colon tissue, and on the right side, a high magnification. You can see that the epithelium is completely negative, and all mesenchymal tissues are evenly stained, single lymphocytes in the negative epithelium. It's a very, very good immunohistochemical assay, but vimentin stains everything. So what about cytokeratins? Now, that's the same block, and here we stained with an AE1, AE3 pan cytokeratin marker, and you can see that here the epidermis stains beautifully, the colon epithelium, and so forth. The epithelium here, the tonsil epithelium that covers it. And it's a completely different staining from what you saw before with the vimentin. And then another, this is the higher magnification illustrating the same good staining. This is placenta, this is the kidney, this is lung, and this is pancreas, and all the positive structures actually remain positive, even though it's a multi-tissue sample. So there's technically no problem that the tissues are not covered right, or because maybe the lung wouldn't stain well. No, it stains perfectly. And here's another one, left one. You may not have heard of it. Our hematopathologists use it. It's a B and T lymphocyte marker, and it should just highlight that the same type of block shows a very good resolution, even for antigens that have a very focal distribution, like in this population of lymphocytes. And you see this is the tonsil, and you can also see that the spleen itself serves as a tissue because you have an assemblage of immunopositive cells here. So this is the real big advantage of this. So use as a non-slide control. When we started out to do this, we used this circular shape, but the diameter of 12 millimeter, and you can see this on the top images, you see takes a lot of space up on the slide. So we came up with a more longitudinal version of it, and that's what you see on the right side. And the longitudinal version, we placed it here just for the demo in the middle of the slide. And these are actually to the scale. This is 14 by 6 millimeters, and we have the same tissues inserted, just in a different pattern. Advantage you can see here. So if you have a longitudinal tissue, there is lots of space for a rather big tissue on the remaining area of the slide. And you see that, that's a colonic carcinoma, which we kept in this on this slide. So this is just, again, the tissues that we're using, and this is not actually a magnification of this, it's just a general picture of how those tissues, what tissues we are using. So what are the advantages of this? Now, when you normally do IHC controls and you use just one tissue or even one tumor tissue, it's difficult to really show the difference for some of the antibodies that we're using. Here, the demonstration is easy. The tumor tissue shows different types of staining. Here's a CD3 staining. Here is a CK20 staining. This is a cytokeratin staining. And these are the controls. So each of the controls gives a unique staining pattern with the 10 tissues, the 9 within tissues that we're using as controlled tissues. And this is an example that actually again highlights this ability. The H&E shows you one thing, vimentin, that's easy. But look here, this is a pretty identical thing. Why? This is a cytokeratin staining. This is actually, I think, a metastatic tumor. This is a pan-cytokeratin staining with an antibody called OSCAR. And you see that the staining is pretty much similar. So just by looking at the tumor tissue, or let's say you would punch out a tumor area and use it as a control, you will not be able to see the difference between these two assays. But look at the controls. There is only settled staining in one of the structures of this tissue, which is, just the overview, you can see it. But here you see that several of those are positive. So what our control does, it actually doesn't only control the IHC assay, it also tells you which antibody has been used. And that's what puts the safety of IHC on a completely different pattern. In the course of doing these carrier-based multi-tissue block, we also develop other antibody, other multi-tissue block shapes. The one we most commonly use right now is this one. The only difference between this one and this one is that we leave the spleen just as it is. Why? Because this is our to-go-to multi-tissue block when we test a novel tissue, a normal antibody. So when we test a normal antibody, that's what we use, because we don't use it as an on-slide control. We can use the space of the whole slide in order to test a new antibody. The carrier system we also use when we have very small single controls. For example, if you have mutational antigens and you have a protocol that is only positive in one tissue, we will punch out a very small diameter also of a cell pallet or of a mutated tumor and also place it in the carrier spleen. Why? Because it's much easier to section. It's much better visible on the slide, and you can get away with much smaller amount of control tissue. This is another one to the right side here, which we call which we use in order to use huge-sized tumor samples. This is a colorectal multi-tissue black of 5 tumors. Why do we place them in a carrier? Because they're much easier to cut. Each section is pretty big, and when we want to see if our newly established antibody is positive in a certain type of tumor, like colorectal tumor, and we want to test 20 colorectal cancers, we use 4 of these blocks with 5 tissues each, and have a very small amount of blocks to section, of slides to stain, and still know what the new antibody does in 20 tissues. And finally, I don't know if you can see this well, we also try to do a TMA and use the carrier tissue. And in this case, we used liver. And it works just as beautifully. The procedure is always the same. Just like with this one, you have to re-embed it. But Remember, when you do a TMA, normally the problem is, even with you have a new machine like the new tissue arrayers, once the cores are into paraffin, you cannot really melt the paraffin. You can only heat it a little bit, push it down, and so forth. But still, when you section it, even with the newest equipment, it feels like you do many, many different cuts and sections through many, many different tissues. If you place all the cores of a TMA in a carrier tissue and melt it completely and let it solidify again, what we call re-embedding, you will see this cuts like one section, and you will not lose any of the punches of the cores that are in those TMAs. Taken together, the carrier approach is a very good way in order to assemble several tissues in a multi-tissue block. It's very easy and fast to generate. The technicians in our lab are now well versed. If you have all the tissues handy, and we have a whole collection of these tissues that we use over and over again to make new of these multi-tissue blocks, normal tissue multi-tissue blocks, it's very easy to punch them out. You do one of these blocks in maximum 10 minutes. And if you do 5 at the same time, you further cut down. And the nice thing is that the carrier allows for an exact placement of the tissues that you normally have to be upright. For example, skin, colon, everything with a surface is much better to be sectioned and handled in a carrier tissue. You can reduce the size of the tissue, and you can reduce the size of one block. The carrier also ensures that the section and trimming of a block is much easier, especially even applies to your TMAs, which we rarely use. But try, if you do TMAs, use a carrier tissue and you will see a big difference. Re-embedding is the key thing. The handling of the section is also easier because when you section them, put them in the water bath, the loss of cores or punches is minimal. It never happens to you. If you ever work with other multi-tissue blocks, oftentimes you lose some of the cores when they're placed in the water bath and then when they go from the water bath onto the slide. And what's the advantage of taking a normal tissue multi-block to test new antibodies? It's basically you can use one block for 80% of your IHC protocols, whether it's CD markers, it's keratins, you all use the same block. Each IHC protocol will render a typical, very peculiar staining on those tissues that I mentioned, which we are using. The safety of the IHC protocol will be enhanced because you actually know which antibody was used, and you do not have to rely on that the right control was used. And also, the staining can be controlled over time, even if you change the different skins, livers that are in there. Overall, a certain protocol will always generate a very similar staining. And it's a very sensitive marker that immediately shows that something also is wrong with the antibody that we're using. We often have this. We just had it 3 weeks ago that one of our multi-tissue block instantly showed, oh, there's something wrong with this antibody, and it turned out it lost reactivity. We had to actually switch to a different clone. And eventually, it will help to streamline the processes in your lab. And because you can use the same tissues that you initially use to test the new antibody as an on-slide control in the downstream application. Nothing works with the people actually doing this. The multi-tissue blocks and everything is done by Denise, who is the senior tech in the lab, to the left side, Emily and Jerica. And it's Denise who is working with me for a long time, and we both developed this technology. We also have, our institution has applied for patent protection, so don't do a company on this idea. Memorial will come after you. So finally, I would like to say thank you. If you have any further questions about this, there's my email address, which is correct. And I would like to say again, thank you to Diapath and its whole team for inviting me for this presentation.
Okay, many thanks, Dr. Jungbluth, for a very interesting presentation. And now it's time for discussion, and I will break the ice with a few questions myself. The first question is, as you said, the normal tissues are ideal for initial testing of the antibodies, and you have developed your multi-tissue block for this purpose. What are the reasons? You explained it a little bit in your presentation, but I want you to stress it. What are the reasons to prefer normal tissue over tumor tissue as internal quality controls for routine immunohistochemistry testing?
The idea behind this is that the expression of most proteins is very consistent in normal tissues. While when you look at tumors, you know, we all know you stain certain types of tumors, and it's usually several markers that you assemble to make a diagnosis. But let's stick with, because you know that's really my background, but you could use any other tumor in many different antigens. Melanomas. You know, I worked with maybe 5 different melanocyte differentiation markers. That's how we call it, the markers that are positive in melanomas. There's, of course, GP100, which is the antigen that's detected by HMB45, the classical antibody to melanomas. Then you have melan A, which the antibody is detected by the clone A103. Then there is tyrosinase. It's an antibody, it's less known in the field, but it's a very sensitive marker. But all of these markers are not positive in 100% of the melanomas. So if you have tough luck, and you have and they also show heterogeneity in the tumor. So you know that some tumors show HMB45 staining only in certain areas, and that applies to melan A as well. So for example, if you have a control, the expression may vary depending what melanoma you're taking. Also, you can do deeper section, and all of a sudden the melan A expression goes down and the staining will go down. But if you take a normal skin, I promise you there will be definitely 4 to 5 positive melanocytes with any of these markers. May not be the same one, which already tells you that. But it's a very sensitive method. If you see in our multi-tissue block melan A staining positive in the skin, that's all you need. It's a very sensitive way to do this. On top of it, when you assemble them in the multi-control, each of the antibodies gives a peculiar staining pattern. You know, you can do 50 different stainings on the same multi-tissue block, and you can tell with some practice which antibody was used. If you used control tumors, you have to always rely on what's the right control used. Is it the control still good? But if you do a multi-tissue block, you can actually say, wow, this is this antibody. And you know, sometimes, especially if you work a lot with concentrated antibodies and you do your own dilutions, confusions can happen. And with our technology, you instantly see, oh, this was the wrong antibody. The control doesn't match what I was actually ordering.
Yes, and so you provided with a lot, even with a lot of negative controls at the same time as the positive control of
the negative controls are just as important as the positive controls. Yes.
Okay. Second question. I want you to give us more information about these little pen manual devices you've been using to obtain the right carrier portion of tissue and to extract, discard, or insert into the holes of the carrier tissue, the donor tissues.
Yes, these colloquial called here in the US derm punches. They're actually used by dermatopathologists to extract nevi. They're very sharp. They don't cost a lot. You can get them through the usual providers for medical equipment. Here, bigger companies in the US, Fisher. I checked you, even Amazon, you can get derm punches. I don't know why you would order them on Amazon, but I'm not advertising to use them, but you get them there. You just put in derm punch, and you get them in different sizes. The normal sizes that we use varies on the application. For our normal tissue, you could see we use 3 to maximum 4 millimeters for colon and skin because we want to have a little bit more of the surface. And for the other homogeneous tissues, for liver or pancreas, it's fine if you take 2 millimeters because it shows all the structures. With 2 millimeters in the liver, you always have a portal field, you always have a central vein, and most of the time you have several of those. So, and they're very sharp. You have to, from a technical standpoint, to exchange them. You cannot use them for 3 of those multi-tissue blocks, but they are cheap. You just discard them. And when you punch out the paraffin, usually the core stuck into this device. And we have a small needle that you normally use in the kitchen to test whether your cake is done. It was our cheapest lab equipment. It was 75 cents when we bought it. And it goes right through the punch device from the back, which is hollow inside. And you push out the core. And the core, normally when we extract it from the donor tissue, we just put the donor on top of the hole of the acceptor and just push it into the hole. And you get also of derm punches that have a device inside that basically is like when you do a pen, you push on the button and then there's an inside device that pushes out the core. They're a little bit more expensive, but much more convenient to use. And you get them in all sizes.
As far as I understand, is the diameter of the hole in the carrier tissue the same as the diameter of the core you produce of the donor tissue, or
yes.
Is it bigger? It's just the same.
We use the same size because you wouldn't get them in a size that they just fit, neatly into this acceptor hole. Also, if you take one, you get them almost in increments of 1 millimeter, 1 millimeter, 2 millimeter, 3 millimeters, and that's too big. So when you insert them in the carrier tissue, when you insert the donor punches into the carrier tissues, it looks horrible because there's space. They would juggle. You cannot use them. You have to re-embed them. So they juggle inside, and even if they would fit in nicely, the re-embedding does the trick for the sectioning. So the carrier has several advantages. It holds it in place, even if you can't believe it after you've done your first one, say, is this going to work? But you put a mold on top when you've done everything inserted, then you turn it around and put it in the oven. And then you let it melt for an hour or 2 at the melting temperature, at the melting point of the paraffin. And the only thing you do is you take the cassette off from the mold and push down gently the carrier tissue and everything that's inside. And the tissue I showed you is exactly from doing this. And then you put a new cassette or the old cassette on top of it, fill up a little bit of the paraffin that the cassette holds it properly, and then you put it in the fridge. When you walk with a thing through lab, don't, say it, keep it safe, put it in the refrigerator and let it cool down slowly overnight. Don't get anxious and say, I want to see what happens. Don't pull it out overnight. If you leave this in for a couple of hours, you easily get the block out, and it's the most beautiful thing you have ever seen when doing embedding of tissues for maybe the last 10 years.
Okay. So now two more questions. Do you think that it would be possible to standardize the kind and number of normal tissues assembled in a universal control multi-tissue block for internal quality control of more than 80% of the antibodies normally used in a lab? And do you think it would be possible to make it universally used and possibly to develop an automatic control of each immunostained slice produced through an artificial intelligence program before devising the slice to the pathologist?
So this is actually the third aim that we had when doing this, because we were seeing that, there are so many protocols in the lab, and quality control right now works with an experienced technician looking at the control tissue and saying, oh, this is a good assay or not, or this worked, or we have to repeat it. And you know, I think Memorial Sloan-Kettering Cancer Center and our Department of Pathology is the most active in digitizing slides. We have a huge slide scanning capability. And it's only a question of time when all IHC will be scanned, not only in our department, but in general. We are going through a lot of changes in pathology right now. And the evident question is, can't we put IHC on the next level? And that's exactly our idea. What you asked is, if we always use the same tissue, can't we use artificial intelligence to teach a software how to interpret the control tissue, especially since the control tissue always consists of the same tissues. If necessary, we can place the skin always in the same spot, in the colon and so on. But according to the people that we taught from AI, that's not even necessary. AI systems are actually able to recognize what tissue is in which place. And yes, that's our aim, and we're actually working on it. And again, that's why we protected, or that's why our institution protected this with a patent, so that nobody can use it. You can use our ID in your own lab as a control as long as you want. But if you want to make a company and came up with an AI algorithm how to read it, our OTD department may send you an email and say, look, don't do it. But that's the aim, because sooner or later, I think in 10 years, everything is going to be scanned, also the IHCs. And the question come up, so when we scan the slide anyways, can we do a quality control? And yes, I think this would be ideal for quality control in the future.
Yes, and the ideal would be to be, in a way, forced to make very good controls for immunohistochemistry, because so far there are a lot of advice. There is a lot of advice, but you still left to a certain degree of local decision.
Yes, the interesting thing is also when we use, if you would use our multi-tissue block with the same tissues in your department and we would use it, we could actually compare our staining across different institutions. We could also, I can tell you, it's a very sensitive marker. We can tell instantly if an antibody, the reactivity is down, because we know when we used it in the same tissues, the previous lot of a certain clone, all of a sudden the reactivity comes down. We had this problem I alluded to with one of those checkpoint molecule markers recently, not the big ones, but there are different of these co-stimulatory molecules. And we instantly saw, boom, there's something wrong with the antibody. And we eventually had to buy a new, to test a new clone, because the same clone across different manufacturers showed the same problems. It's very sensitive to, as a longitudinal control, not only for the control of the tissue that's on the slide. And that's actually what we're working on right now.
Which is really good. Then the last question is, you to assemble your multi-tissue block, you use normal tissue, which is kind of leftover from normal human
procedures,
reaching your lab, and we all do it. But have you tried to compose a multi-tissue block with animal tissue?
We've worked with animal tissue in the past. The problem, though, is animal tissue is not completely identical to human tissue as the genes. It depends on how far they're evolutionary preserved. If you go on, for example, certain proteins, cell cycle markers, CDK molecules, they're pretty much the same across. You can test yeast and you will see positive IHC. But if you get, for example, in later evolutionary antigens, it's that you have problems because the identity of the proteins is not 100%. And of course, you never know which is identical or not. You have databases where you can compare to them. You can compare the different markers. But then again, you would end up with a control if you use tissues that would work for some antibodies, but not for others, because this protein is different in pig from human, but the other ones you can use. So it would add a lot of complexity, and many things may distract from the actual aim to make it consistent. It would be inconsistent for certain antigens, but not for others. I think it would create more confusion than it would create help.
Yeah. Okay. Thanks a lot.
Thank you for having me again, and hope we talk again.
Okay, we will talk again, I'm sure.
Yes. Bye. Thank you very much, Lauren.